Hey, I'm Andrae Manglo, together with my partner Cedric Phillip Quizon, is here to present our paper entitled A Review Paper on How Ultraviolet C Irradiation Affects Post-Harvest Tomato.
The objectives of the article is to determine and discuss the effects of UVC irradiation on the gene expression, bioactive compounds, and morphological quality of post-harvest tomatoes.
Tomatoes suffer from quality degradation and have a shorter shelf life due to significant post-harvest changes. The buildup of reactive oxygen species, microbial development, cell wall degrading enzymes, and a faster respiration rate are the most noticeable changes in tomato fruit that contribute to a quality loss. The development of cell wall disintegrating enzymes such as pectin, metylesterase, polygalacturonase, and cellulase causes a significant loss of firmness. As a result of spoilage or pathogenic bacteria, including Rhizopus stolonifer, Alternaria alternata, Botrytis cinerea, and others, microbial growth leads to deterioration. One of the growing non-chemical and non-thermal methods of post-harvest procedures in extending the shelf life of horticultural goods is UVC irradiation. UVC irradiation was first utilized as a non-thermal method to minimize microbial development in liquid meals and to contaminate fresh-cut fruits. Also, UVC irradiation has been shown to induce disease resistance in sweet potatoes and develop phytoalexins in orange and carrot tissues. This treatment was proven to slow the ripening of climacteric fruits, including apples, peaches, and tomatoes. Phenolic compounds. Phenylpropanoid molecules are produced by plants as part of their defensive mechanism. Some phenylpropanoid molecules, for example, may act as UV protective shields as an antimicrobial chemicals like phytoalexins. Lignin derived from phenylpropanoid polymers may operate as an inactivating physical barrier for pathogen entry. Therefore, phenylpropanoid metabolism plays a key role in plant defense. Phenylalanine ammoniase is known as the key enzyme in plants that regulates metabolic flow from primary to phenylpropanoid metabolism. As a result, the regulatory mechanisms governing the PAL gene have been widely investigated in terms of their induction by a variety of stressors, including ultraviolet light injury, pathogen infection, and elicitor molecules.
The study of Maharaj et al. To assess the effects of UVC on post-harvest tomato, mature green tomato fruit were subjected to different doses of UVC. For the first week, high level of total phenols were seen in UVC-treated fruits. They induce fast cell death with necrotic morphology, but it results in the fruit not ripening properly and turning browns in color. The result indicates that the sample treated with a UVC dose of 3.7 kJ per square meter could be utilized to increase total phenolic content and improve the tomato quality. In the study of Bravo et al., the UVC treatment significantly induced the total phenolic content of the tomato. After 12 hours of exposure to UVC light, the phenolic content increased by approximately 42%. This may be due to the phenolic biosynthesis pathway because the UVC treatment may cause the accumulation of phenolic compounds in the sample. In the study of Liu et al., they used a mature green tomato fruit subjected to different UVC irradiation. UVC irradiation significantly raised the total flavonoid content of the tomato fruit after 35 days of storage according to the result. Furthermore, tomato fruit irradiated with 16 kJ per square meter had the highest total flavonoid concentration after 35 days of storage. Moreover, in the study of Rabo et al. States that the accumulation of the DNA damaging effect the ultraviolet C irradiation treatment may increase the ultraviolet absorbing that may absorb the flavonoid and other bioactive compounds. These effects may be due to the activity of enzyme called phenylalanine ammonia lyase, which is a key enzyme that is connected in producing phenylpropanoids. In the study of Magwaza et al. States that exposing tomato fruit to UVC for 60 minutes may enhance antioxidant activity. Similar results were observed on the irradiated mature green tomatoes in the study of Sergio et al. The antioxidant activity of treated tomato was substantially higher than that of untreated sample after UVC irradiation treatment. The lipophilic antioxidant activity of the tomato fruit increased after 1 hour and 3 hours of ultraviolet C irradiation treatment. The treatment positively affects hydrophilic antioxidant activity, but the positive impact is entirely reliant on the hormetic treatment condition.
Tomato samples can inhibit microbial growth, slow degradation, and delay senescence. In UVC ripening tomatoes, cell wall degrading enzymes such as polygalacturonase, pectin methylesterase, cellulose, silanase, degalactosidase, and protease were shown to be reduced. However, it was revealed that the UVC treatment did not affect the sample fairness of ripe tomato fruits based on the studies conducted by other researchers. To further analyze the morphological changes of post-harvest tomato fruit under the UVC irradiation treatment, the following sections or the next sections will discuss the changes by layers of the tomato pericarp. Tomatoes undergo several adverse alterations during post-harvest storage. Pathogen-caused illnesses such as fungus and bacteria, decreased antioxidant activity, early fruit maturation, and reduced post-harvest lifespan are factors that contribute to the decline in fruit quality after harvest. The primary post-harvest issue with tomatoes, like with other fruits and vegetables, is excessive softness, loss of turgor, and the degradation of starch or the breakdown of the fruit cell walls caused by a fungal attack could cause the fruit to become soft. UVC has been demonstrated to dramatically suppress the activities of the pericarp cell wall degrading enzyme cellulase, polygalacturonase, and the pectin methylesterase, leading to increased firmness retention. Ripening of climacteric fruit such as tomato is primarily controlled by ethylene production, which initiates a series of biochemical and physiological processes that causes textural softening. While the process governing variations in firmness throughout ripening are not completely understood, softening of tomato fruit flesh in storage has been linked to changes in the pericarp wall, notably the degradation in the middle lamellae pectins. Various tissue make up the tomato's pericarp, which is separated into exocarp, mesocarp, and endocarp. Exocarp cells had demonstrated to keep their cellular stability longer than mesocarp cells throughout the senescence process. This was shown by less degraded organelles such as chromoplasts, which still had the plastoglobuli in the exocarp cells but were degraded to multilamellar membrane structures in the mesocarp cells. Exocarp and the outer mesocarp cells in the untreated fruit exhibited change despite the absence of pathogen hyphae in the tissue 2 days after inoculation in the inoculated control fruit, while the cuticle remained intact. The underpinning cell wall was wavy and swollen. The exocarp cells were extensively vacuolated, and electron-dense substances were found in a few of the vacuoles. While In the UV-treated tissue 2 days after being inoculated, there was no signs that the fungus had caused any harm. The fungus was not present in the tissue and no changes to the cell wall were apparent. The vascular bundle included parenchyma cells with complete organelles. At a comparable time, the cell walls of the paratracheal parenchyma cells were much more stained than those of the inoculated control. Firmness or researchers, UV irradiation had no effect on the color of ripe tomato fruits, while other researchers claim that UV irradiation after harvest was often less efficient in altering tomato color, which seemed to be mostly determined by the harvesting stage. Color parameters found to be altered more significantly for flesh than for peel. Weight loss. Irradiated fruit lost less weight and had slower color change and softening than non-irradiated food. The decrease in fruit weight loss was more pronounced in fruit treated with high UV intensity. This might be linked to an enhanced tissue integrity mechanism. However, a low dosage of UVC irradiation could result in greater weight loss. Researchers discovered that exogenous polyamides inhibited cell membrane weakening and polygalacturonase activity. Several years ago, it was investigated that polyamides work in the same way that calcium did. This means that the cell wall would be more difficult for enzymes to pass through. Moreover, lipoxygenase or LOX, a non-heme iron-containing dioxygenase, helps in adding molecular oxygen to fatty acids that have a cis or cis-1 4-pentadiene system. This leads to the formation of hydroperoxide or HPO derivatives, which have been linked to stress responses, wounding, and pathogen attacks. It was found out that increased LOX activity has been linked to the loss of firmness of fruit during ripening.
This review paper discusses the various effects of applying UVC irradiation treatment on post-harvest tomatoes. It highlights the impact of UVC irradiation treatment on gene expression, bioactive compounds, and morphological changes. The UVC irradiation treatment causes changes to the gene expression of the tomato and can generate an enzyme activity and gene expression including phenylalanine ammonia lyase. The UVC radiation treatment also lead to a large-scale alteration in the tomato bioactive compounds. The total phenolic content of the tomato increase after UVC irradiation. Employing UVC irradiation triggers and enhances the level of flavonoids. It also shows a significant effects on the antioxidant activity and lycopene content. The study also showed that UVC irradiation has a beneficial impact on the morphological quality of tomatoes. UVC treatment can reduce firmness loss, weight loss, and color development of tomatoes during storage and handling. This can help the tomato to have a much lower deterioration and ripening, making UVC treatment an important post-harvest technique. For recommendation, other researchers may focus on developing cultivar-specific UVC irradiation techniques for optimal tomato fruit preservation, since the treatment time and cultivar have also been demonstrated to affect color development, firmness, and weight reduction. Combined non-thermal technology can be used as a post-harvest treatment on tomatoes to improve the shelf life and can help to boost the some bioactive compounds. The future researcher can also use the combined and thermal technology, which is the ultrasonic energy and UVC irradiation, to determine its effects of it on gene expression and morphological changes on a post-harvest tomato.